Cyclins are among the most important core cell cycle regulators. They are a group of related proteins, and the most basic ones typed in humans are G1 cyclins, G1S cyclins, S cyclins, and M cyclins. As the name suggests, each one of these helps a particular phase and helps drive the events of that phase or period. In order to drive the cell cycle forward, a cyclin must activate or inactivate many target proteins at a cell. To do this, the cyclin partners with a family of enzymes called cyclin-dependent kinases. Alone CDK is inactive, but binding with the cyclin activates it, making it a functional enzyme that's able to modify the target protein. The cell cycle is a four-stage process consisting of the G1 phase, where a cell increases in size and the cellular contents are duplicated, the S phase, where DNA replication occurs and each of the first chromosomes are replicated by the cell, the G2 phase, where the cell prepares for cell division, and the M phase, where mitosis is followed by cytokinesis occurs with the formation of two identical daughter cells. There are three checkpoints in the cell cycle. The first is the G1 checkpoint. Here they check for the cell size and that there is no damage to the DNA, so it will be allowed to go to the S phase. Next is the G2 checkpoint. Here it ensures the chromosomes have been replicated and that the DNA is not damaged. And finally the M checkpoint. Here where citric chromatids are correctly attached to the spindle microtubule.
The chromatin fibers become more tightly coiled, condensing into chromosomes. The nuclei disappear, and each duplicated chromosome appears as two identical sister chromatids joined at the centromeres. This meiotic spindle begins to form. It's composed of the centromeres and the microtubule. The centrosomes move away from each other, propelled partly because of the lengthening. Chromosomes are now at opposite poles of the cell. The chromosomes convene at the metaphase plate, a plane that's between the spindle's two poles. The chromosome centromeres lie at the metaphase plate. And for each chromosome, the kinochores of the sister chromatids are attached to the kinochore microtubules coming from the opposite poles. Anaphase is the shortest stage of mitosis, and anaphase begins because cohesion proteins are created. This allows two sister chromatids to part and become chromosomes. The two liberated daughter chromosomes begin moving to opposite sides, and the cell elongates because the two microtubules lengthen, and by the end of anaphase, the two ends of cells separate into equivalent and equal collection of chromosomes. Two daughter nuclei form in the cell. Nuclear envelopes arise from the fragments of the parent's nuclear cell. The nuclei reappear, the chromosomes become less condensed, and the any remaining spindle microtubules will depolymerized. Mitosis is now complete. The division of the cytoplasm is underway by a late telophase, so the two daughter cells form shortly after mitosis. In animal cells, cytokinesis involves the
Cancer is the process where cells undergo uncontrollable division. The process begins when a normal body cell undergoes transformation into a cancer cell. Typically, division is limited either by anchorage dependence on a structural substratum or by density-dependent inhibition, where crowded cells are kept from dividing further. As the cancer transformation occurs, new cancerous cells are deposited to a tumor. Tumors fall into one of two categories, benign and malignant. Like their name implies, benign tumors are often harmless masses of cancerous cells. They have too few genetic and cellular changes to survive outside of their original tumor sites, where they remain. Benign tumors can be easily removed with surgery if the patient desires. Malignant tumors are polar opposite of benign tumors. These are dangerous deposits of cells, altered to the point that their genetic changes allow them to survive outside of the original sites. These tumors take advantage of metastasis, where some cells separate from the original tumor, enter the bloodstream, and travel to form new tumors in distant parts of the body. Metastasis makes malignant tumors serious life-threatening issues with no easy treatment.